Carly Romano, and this is breaking news. A scientist, Emma, at a local lab has misplaced the sugar concentrations of the solution. Two scientists at the local lab have taken on the task of trying to figure out the different sugar concentrations.
Thank you, Carly. Speaking of which, I think I see some scientists right now.
Oh, did you see them? Oh my goodness. I'm sorry,
This is such a surprise.
we can't talk that long because we have to get our materials ready. And we need potatoes, a beaker, knives, cork borers, and balances.
But we can't forget petri dishes, a camera, and a graduated senator.
Yes, but we need to set up. So we're going to head out and set up.
All right, well, we'll see them in a little bit right up in the lab. Back to you.
The scientists sent me their hypothesis this morning. I'll read it out loud for the viewers. If the potato expands, it is hypotonic. If the potato wilts, it is hypertonic. The potato that expands has been given the highest concentration of the sugar solution.
Welcome back, viewers. The scientists have headed into the lab where they will perform their experiment. Right now, I will remain out while the camera crew goes in and watches how they perform this. It's not a fry.
Yes. Just a moment. I'm sorry. Thank you. Oh, sorry.
Heck yeah.
A little thingy.
Okay.
How many do we
We
have?
have 7 here.
Conduct this experiment. We have to find out what the measurement and mass is of each potato wedge. So right now, I am going to turn this on, have the mass set on. I'm going to drop one of these, and I'm going to measure it before the experiment. So this is exactly 1 gram. Test subject 1 is 1 gram, 1 inch. Well, they're all 1 inch, so this one's 1 gram. The second one is exactly 1 gram as well. The third one is also 1 gram.
That'd be great if all these 1 gram, we don't have to throw all this out.
1.1
Oh
gram.
my gosh.
We can see if we can cut it a
I
little
think
bit.
they mind.
Hold on.
It's fine.
1.1. So let's see if any of these are different. This one's 1, so we'll replace this one with this one.
Which one is
The
that?
fourth one with 1. And let's hope this is 1 as well. 1.
Thank you.
So
Thank you so much.
those are discarded. These are not the right ones.
Oh, I didn't see you there. Did you see them?
I did not.
Well, let's go ahead and explain exactly what we're doing in this laboratory.
Well, today we will know which one has the highest concentration of sugar solutions.
And how exactly will we do this? Well, we will be doing this by putting each potato, here Ashley, let's put each one inside.
All right.
Put each one inside, and in 24 hours, we will observe exactly what happens and when one wilts actually, what happens, and what happens when it expands.
When it expands,
Isn't that hypotonic?
it is.
And we'll see you in 24 hours.
Wait, wipe them off. Here you go. I see no change. Okay, let's put this in. Should we rinse this off? I'll rinse it off.
As you can tell right here, we're going to be measuring the before and after. So we already did the before, and now we're going to do the after. So Ashley's going to come up, and she's going to specifically look at the weight. So from the purple,
So, as we can tell, this is exactly 1.3 grams, which originally it was 1. So, obviously, it has a heavier mass than before.
and Victoria over on this side is going to be recording each data, the weight, and observation. And now for the blue.
Wow, this is a whopping 0.6 grams.
That dropped 0.4.
That's crazy. That's even a bigger difference than before.
Now let's see what the other ones are like. This is going to be green.
So it only dropped 0.1, but that is still a significant difference.
Any difference can mean a difference. Oh, Ashley, can you repeat what that one was? I think it was 0.9 specifically.
0.9 grams.
Now this is the yellow. Ooh,
Oh, well, this one rose up to 0.1, which is
kind of the opposite of what green was like, right?
exactly.
And
Yellow
now
is
our
completely
very
the opposite.
last one is going to be the red. Now you may see specifically what this one is. Here, zoom in specifically on that one right here.
Oh, well, this one also states that it's 1.1 grams, which is a 0.1 gram difference than before.
So over here, we're gonna hit it off. Back to you guys.
Hello, welcome back. We are here with another group from California to see their results on their enzyme experiment. I have one question for you. Do you consider your experiment a success? Why?
I think for sure ours was a success because when we were able to put the concentrations in order, it was in the correct order as the matching solutions. So I feel that our experiment was correct as it matched the actual plane experiment.
That's great. I also agree. Our experiment we were able to weigh the masses of the different potatoes after they were placed in the concentration to see which one was more concentrated in sugar. All right, and did you weigh the sample potatoes before and after the experiment? If so, what was your results?
So what we did for our experiment was we weighed the potatoes before and we got all of them to equal 5.7 grams so that we could ensure that the results would be the same for our control group and for all of the other ones. But then we weighed it after to measure how much volume had come into the potato or gone out of the potato. But what we also did was measure volume of the glass before the potato was inserted and after to see if there was a difference in volume, and that's how we were able to also tell if it was a hypertonic or hypotonic or even isotonic solution.
That's amazing. Our group also did something along the lines of what your group did. We made sure there's a balance, a control between the weight and length of the potato. So our control, we started off with a 1-inch potato and they all weighed 1 gram. And after they were submerged in the solution for 24 hours, we were able to compare which one weighed more and which one stayed the same to determine which ones were hyper or hypotonic. Back to you, Carly.
Welcome back. We will now talk to the brave scientists that took on this daunting task. Ashley and Hope, welcome.
Thank you.
Describe your experiment as a whole. What happened? Was it a success?
It was a complete success.
Complete. I don't know how else to describe it. Everything went absolutely perfect. We found out that purple was hypotonic.
And blue was hypertonic.
And what was yellow?
Isotonic.
At least they're making money and they got their doctorate degree. Viewers, here's a graph to show their findings.
Okay, so for those of you at home, this is the chart that Hope and Ashley were talking about. So this is the order of concentration low to high for the solutions. So it went purple, red, yellow, green, and blue. So the weight before for purple was 1 gram, the weight after was 1.3 grams. For red, it was 1 gram, and the weight after was 1.1 gram. For yellow, the weight before was 1 gram, and the weight after was also 1 gram. And then for green, the weight before was 1 gram, and the weight after was 0.9 grams. And then finally for blue, the weight before was 1 gram, and the weight after was 0.6 grams. So just some quick observations. For purple, it was cold to touch, and it was the hardest potato. For red, it was harder than the purple. For yellow, it was hard but stayed the same. Green, it was firm but also soft. And for blue, it was squishy. So the water potential for purple was 0. For red, it was negative 4.92. For yellow, it was negative 9.84. For green, it was negative 14.76. And for blue, it was negative 19.68.
Now, can you guys go a little bit more in depth as to what happened?
Yes. So the purple was hypotonic because in the beginning weight it was 1 gram looking at the chart that was just given, and then it went to 1.3. So that meant that the cell absorbed water, which is hypotonic, and also the water potential was 0. So as you can tell, it was hypotonic. And then Ashley's going to talk about the blue one.
So the blue one, actually it lost 0.4 grams of its weight, and that is due to it being hypertonic. That means that the concentration travels from high to low, and the water is actually pushed out of the cell, in this case, the potato.
And what was the water potential for blue?
Oh, it was negative 19.68.
That's a big difference from what hypo is.
Oh, definitely.
But yellow was more of an isotonic, so it went from 1 gram to 1 gram after spending 24 hours in the solution. So what the water potential in the yellow was equal to the potato, and so that's why we know that it's isotonic. It's equal.
Yes.
Equilibrium. Yeah,
Yes.
you're right.
Equilibrium. All right, wow. So were there any potential errors in your experiment?
I mean, we're human, so there's always going to be errors. Yes, we had different things that we could do.
Yeah, probably the water concentration wasn't
Measured,
exactly measured correctly.
Cutting the potato. Well, we were doing that by hand and with a ruler, so when you go down on the knife, it could slip and you'd lose maybe like 1/16 of a potato. So that could definitely be a factor, I think.
Yeah, and the grams, the weight of
Yeah.
it, maybe the scale was a little bit off.
Yeah, it was off until we weighed one again.
That is true.
That was yellow, so we thought yellow was 1 and then 1.1. So that would have altered what we thought, which would have been equivalent to red. But we measured again because we knew something was a little bit off. So there are always errors in a lab.
All right, well, thank you guys. Thank you guys for tuning in at home. Tune in tomorrow when we talk about the rate of enzyme reactions. This is Carly Romano signing off.